Hello students, today we shall continue chapter 6 and our today's topic is search for the genetic material. In this topic we shall discuss different efforts done by many scientists. So first of all we will discuss the Griff's experiment for transforming principle. In 1928, Frederick Griff he did an experiment on bacteria which causes pneumonia. In his experiment, he observed that there are two types of bacteria which were responsible for this disease. The bacteria is Streptococcus pneumoniae and it has two types of strain, S strain and R strain. The S strain bacteria, they produce smooth colonies. Why? Because their cell wall has a coating of some polysaccharide, and R strain bacteria lack that coating, so they produce rough colonies. So S strain bacteria are virulent, means they are able to cause disease, and R strain bacteria are non-virulent, they do not cause disease. So in his experiment, Griff did what? He took S strain bacteria and filled in an injection and then injected into the body of a healthy mice. After some days, he observed that the mice died. When the samples were collected from the body of the mice, S strain bacteria were present in the body of mice. Means that S strain bacteria caused pneumonia in the mice and mice died due to pneumonia. Now when he injected R strain bacteria into healthy mice and mice survived, means the R strain was not able to cause disease. Now he took the sample of S strain bacteria and strongly heated them. So we know strong heating kills the living cells. So now he took the extract of that S strain heat killed bacteria and he injected into body of mice. Now this time he observed that the mice survived. Okay, it means the heat killed S strain bacteria were not able to cause pneumonia in mice. But in another experiment, now he took the extract of S strain heat killed bacteria and mixed with live R strain bacteria and injected into the body of mice, and this time he was surprised to see that the mice died. When the samples were collected from the dead body of the mice, live S strain bacteria were found in the body of mice. So where from the S strain bacteria came? So Griff explained that there was something in the heat killed S strain bacteria which could not be destroyed by heating, and that converted R strain bacteria into S strain bacteria, and they could cause disease in the mice and mice died. So he called this as a transforming principle. There is some transforming principle working behind the conversion of S strain bacteria into R strain bacteria into S strain. But he could not explain what is actually the material behind this transforming principle. The later studies will reveal this fact, that what was actually transforming principle. Now let us discuss the biochemical nature of transforming principle. Though Griff predicted that there is some transforming principle behind conversion of R strain bacteria into S strain, but he could not explain the biochemical nature. It is the work of three scientists from 1933 to 1944 that proved that what is actually transforming principle. These scientists were Oswald Avery, Pauline MacLeod, and Maclean McCarthy. Actually, they purified the biochemicals from extract of heat-killed S-strain bacteria in the form of protein, RNA, and DNA. Now they did experiment on mice. They took the extract of heat-killed bacteria, purified, and they mixed some R-strain bacteria in it. But before this, they treated that extract with the protein digesting enzyme proteases. It means all the proteins which were present in that extract, they were digested by the proteases, so they were not available. When this mixture was injected into the body of mice, the mice survived because the protein could not convert R-strain bacteria into S-strain bacteria. Now, next to that, they treated the extract of heat-killed S-strain bacteria with RNAs. So, RNA is the enzyme which digests RNA. It means it will break RNA and deactivate it. Then they injected that extract into the body of mice with R-strain bacteria, and this time also mice survived. It means RNA also could not convert R-strain bacteria into S-strain. Now they did what? They first treated the extract from the heated S-strain heat-killed bacteria with DNase enzyme. You know, the DNase enzyme digests DNA. So, this time when they injected this extract in the body of mice, and mice survived. Why? Because DNA is responsible for conversion of R-strain bacteria into S-strain. So, as a conclusion, we can say DNA is the transforming principle of. So, let us first recall it again. When the extract was digested with protein, digesting enzyme proteases, and injected into the body of mice, the mice survived. Died. Why? Because the protein was removed, but RNA and DNA was available. So either RNA or DNA may be the transforming principle. When the extract was treated with RNA digesting enzyme, RNase, again the transformation occurred. It means RNA also is not the transforming principle. But when the extract was digested with DNA digesting enzyme, this time the mice survived. It means transformation could not occur. So by this experiment we can say DNA is the transforming principle, not RNA